Hi, hello, this is Dr. Swapna and welcome to your Learn Lab, your one-stop for learning. We are studying biopsychology, which is BPCC 102. It's an important core subject for BAPCH students of IGNOU. And this one, what we are going to see today, is the last topic of Unit 2 and the last topic of the first block of BPCC 102. And the topic is neuroplasticity. What is neuroplasticity? It is the brain's ability to change and adapt by forming new connections between the brain cells or the neurons. In our previous videos, we have seen how neurons are connected to each other, how impulses travel from one neuron to another neuron, right? So, neuroplasticity is how these connections are formed and how the brain is able to change and adapt. Like after you learn a new skill or maybe after an injury to your brain. When you learn a new language or maybe a new sport or maybe a new musical instrument, the brain literally rewires itself. Why it rewires itself? So that it is able to learn it and be better at the task. Similarly, after a damage or an injury to an area of the brain, either some other part of the brain will take over for the functions, or some of the neurons in that area might form new connections or may regenerate. So all this is what neuroplasticity is all about. Let's look at this amazing function of the brain in detail. Ability of the mature brain to constantly change and adapt is called neuroplasticity. All right. These changes are influenced by synaptic activity, first factor. So we know that synapse is the what? Junction between two neurons where the impulses are exchanged, right? So if the synaptic activity is not efficient, what happens? The signal will not be able to transmit it to the surrounding neurons. A signal coming in will not be spread as it should. So when you are trying to learn something, your brain is generating signals that okay, this learning is happening, right? But the signal is not transmitted to the next neuron efficiently because of what not good enough synaptic activity, in which case your learning is low or not proper or impaired, right? If the synaptic activity is proper, then the brain will be able to send the signal to all the neurons efficiently that see, we are learning something new, be ready, you need to use this, you need to learn this, you need to form new connections, right? So synaptic activity is an important factor for neuroplasticity. Second is genetic influences. What does genetic influences means? It means the influence of all the genes that you have inherited from your parents. All right. So genetic influences can make you, learn things faster, mm, or what? Basically enhance neuroplasticity. Your brain will be able to adapt to changes faster. Okay. Third is environmental influence. A stimulating or an enriching environment enhances neuroplasticity, while a stressful or adverse condition can slow down neuroplasticity. What does this mean? In case you are in an environment which promotes learning, you get regular physical activities, you have a good social circle where you have meaningful conversation and good connection with people, all these enhance neuroplasticity. These are stimulating environments. Then there are environments which lack stimulation, which is like there is no learning environment, it's always stressful, full of trauma, neglect and isolation. All these will have a negative impact on neuroplasticity. Understood? So that was the uh, influence of environment on neuroplasticity. Next, we have injury or trauma. If there is an injury in any part of the brain where the neurons are all degraded or destroyed irreversibly, in that case also neuroplasticity, the mature brain here change and then it finds a new way to adapt to the situation. All right. So that is plasticity, and these are the different factors that influence neuroplasticity. Next, so earlier it was believed that neurons do not divide or they do not regenerate. Once all your neurons are formed when you are a baby, then there won't be any new neurons that are generated in your body. Alright, so if you're losing any neuron, that is a permanent loss. But Recent evidences by scientists have showed that it's not so. Okay, some amount of new neurons are formed, or there is some amount of regeneration happening when the neurons are uh, damaged in adults, right? So, recent evidences point towards neurogenesis in adult. What is neurogenesis? Neurogenesis means development of new neurons. And where are these happening? Most of the development of new neurons happens in two of these areas in our brain. One is hippocampus and the other is olfactory bulb. Hippocampus is a very important part of your brain, which we'll see when we are studying the parts of brain. And now olfactory bulbs. These are nerves which are responsible for your sense of smell. Olfaction means pertaining to smell. Okay, so recent studies have shown that there is some amount of neurogenesis happening in adults and mostly in these two areas, hippocampus or olfactory bulbs. Now, neurogenesis is promoted by, we have seen that, right? Enriching environment and diet. If you have a diet rich in vitamin B12 and all those important minerals, your neurogenesis will be promoted. Remember, vitamin B12 is a very important vitamin required for neurogenesis. Neurogenesis is inhibited by sleep deprivation and stress. You do not sleep enough, you are awake all throughout night, watching something, working or playing, whatever it is not good for your brain because it impairs neurogenesis or repair of the damage, right? And then stress, equally affecting neuroplasticity. Now we have four processes or responses involved in neuroplasticity. So in your exam, the question might come on neuroplastic responses. There are four different types of neuroplastic responses. First one is neural degeneration. Second is neural regeneration. So first it degenerates or gets destroyed. Then it regenerates. It reforms. Third is neural reorganization. It's organized again. And fourth is recovery. Let's look at these one by one. The first one is neural degeneration. What does this mean? It means progressive loss of nerve cells or neurons and their functions. It's not that just the nerve cells are lost, their functions are also lost. Okay. Now, this is irreversible. Often damaged neurons may not be able to recover their functions. Okay. This may be irreversible, not always, it can be. And what causes neural degeneration? It could be glial cells. We saw that we have a type of glial cell which destroys any damaged neuron, right? Immune cells, they can function as immune cells. Sometimes what happens is that these immune cells can, fail to identify the neurons as self cells or these are part of my own body. They may, by mistake, they may, identify these as foreign particles and attack the neurons. And when that happens, there is a damage or irreversible degeneration of neurons. This is not a normal scenario. This happens when there is a mistake in your genes or genetic makeup, wherein the immune cells fail to identify the neurons as a part of their self body, but by mistake identify it as a foreign body and starts attacking it. Alright? And the next cause is environmental factors that we have seen, right? When there is too much of stress, anxiety, or any environmental stimulus which is not good for your brain, all these can cause neuronal degeneration. Then there are degenerative diseases such as Parkinson's, right? Which can cause neural degeneration. Neural degeneration or the degeneration of nerve cells or neurons can happen in two ways. It can be of two types. First is anterograde. Second is retrograde. Let's see what each of these mean. First is anterograde. So here, before that, let me tell you one thing. This is a neuron, right? They have the cell body, we have the axon, and we have the terminal, right? Now, this is not a good diagram, but yeah, this is just for the purpose of understanding. So, the transmission happens in this direction, right? So, we call this is moving forward. If it is, this is backward, right? So, when you look at this neuron, we have two parts. One is the distal part and one is the proximal part. Okay, this is the distal part away from the cell body, and this is the proximal part near the cell body. You need to know these two terms. This term you will need while understanding the structure of the brain also, right? So distal is away from the cell body and proximal is near the cell body. Alright, so when it comes to anterograde, the neuronal degeneration where the axon and its myelin sheath break down after a nerve is damaged or severed. So a nerve here is damaged or cut into two. Okay, now the degeneration, the axon and its myelin sheath is degenerated. It progresses from the site of the cut or degeneration away from the cell body. Okay, if it is cut here, this degeneration progresses away from the cell body in this direction. Okay, distal end of the axon begins to degenerate towards the axon terminal. Distal end means the end that is away from the cell body. It begins to degenerate towards the axon terminal, which was here. This is called anterograde, which means wherever the cut has happened or the damage has been initiated, the damage progresses away from the cell body towards the axon terminal. Next we have retrograde. Now this is exactly opposite of what anterograde was. Okay, here the neuronal damage where degeneration progresses backward from the axon terminal towards the cell body. The degeneration progresses towards the proximal part of the neuron. So if this is your cell body and this is your axon terminal, then this degeneration progresses backward, proximal towards the cell body. Okay, so this was retrograde. There are two types of neuronal degeneration. One is anterograde and the other is retrograde. Okay, that was all about neural degeneration. So we are looking at the four processes, four responses of neuroplasticity, and we saw neural degeneration. And next we have neural regeneration. Let's see what it is. It is the process of repairing or replacing damaged nerve cells either by generating new ones or by restoring the existing ones. Alright, so here a repair happens to replace the damaged nerve cells. How? Either by generating new ones or by restoring existing ones. And we saw generating new ones is called neurogenesis. Which two parts of the brain this happens the most? Hippocampus and olfactory bulbs. Alright, neurons in brain and spinal cord do not regenerate. Okay, there is no regeneration there, which means if a cell is damaged, regeneration means, okay, so this is the axon. This got cut here. Okay, regeneration means from this cut part, the rest of the parts will be made. A terminal will be made and all this happen. This will regrow from here. This is regeneration. Generating new ones means a whole new neuron is formed. So what we are saying here is that inside the brain and inside the spinal cord, this regeneration does not happen. Alright? Neurons in rest of the body can regenerate. So this was all about regeneration. Okay. So how does regeneration happen? It can happen by generation of whole new neurons or just axons, or if a synapse has been damaged, a new synapse will be formed. If the myelin is damaged, new myelin sheath can be formed. So these are the different methods of neural regeneration. How? Either the whole new neuron can be formed or just the axon if only the axon is damaged. If a synapse is damaged, a new synapse will be formed, or if a myelin sheath is damaged, myelin sheath will be formed. Or it can be just repair of existing structures. If it is just a cut, this cut will be repaired. Alright. So, if the myelin sheath is intact, neurons may regenerate and grow through them to their target areas. So we know that there are myelin sheaths, right? and the axons pass through these myelin sheath, or the myelin sheaths cover the axon. If this myelin sheath is intact and only the axon was cut, the neurons can easily regenerate and grow through them to the target areas. Okay, grow back through them and move to the wherever the target cell was. Now, if the neuron gets severed and myelin sheath gets cut completely, no regeneration takes place. Okay, so a myelin sheath has got cut and separated. No regeneration can take place in this situation. If the neuron gets cut and myelin sheath is damaged, new sheath will be formed, but this may not be same as the old one in structure and function. So if a third scenario here is that if the myelin sheath is slightly cut, okay, or it's not that far, just a minor cut is here, something of this sort. In these cases, a new sheath will be formed to cover this up, but it may not be same as the old one in diameter, right? We know that the diameter or the thickness of the myelin sheath can vary. So when the new myelin sheath is put, it may not be exactly same as the old one, which is the only negative side effect of this whole process. All right. So now another important part, collateral sprouts. Collateral, what are collateral sprouts? This is a process. Collateral sprouting is a process where healthy neurons near damaged ones grow new branches to compensate for the loss of function caused by the damaged neurons. So what is happening here? There is a healthy neuron, okay, near a damaged one. This is a healthy neuron near a damaged one. It grows a new branch to compensate for the loss of the function. So yeah, see here. We have two neurons. This got damaged here. This was completely fine. So what it does is, now this is the synapse of the next neuron. Okay. So this healthy neuron will form a new branch so that it can form the synapse to this neuron, which ideally this damaged neuron should have formed, but because it is damaged, it is not able to. Okay. This is what is called collateral sprouting. All right. So this is a process again where healthy neurons near damaged ones. This was the healthy neuron and this was the damaged one. It compensates for the loss of the function by what? What it's doing? It's growing new branch to make up for the function. Okay. Now the sprouting can happen at axon terminal branches, which means if this is the axon terminal branch and if the nearby neuron is damaged, a new branch can form from this axon terminal. Let's use a different color. Yeah. A new branch can form from this axon terminal and connect to the synapse of the next neuron. Alright. Or it can happen at the node of Ranvier. What was node of Ranvier? These were the areas where the myelin sheath is absent. So a new neuron can arise from a node of Ranvier. Okay, so this is what is collateral sprouting, which means the nearby neurons will make up for the damaged neurons. They are friends. If you are not able to do something, you are sick, then I'll do it for you. That's the attitude. This is collateral sprouts. All right. So yeah, we are looking at the four responses of neuroplasticity. We covered neural degeneration. We covered neural regeneration. Now we'll see what is neural reorganization. Ability of the brain to reorganize itself to compensate for the loss of function in damaged areas. This is called neural reorganization. Earlier we see that neurons regenerate to compensate for the loss of the nearby neurons, or new neurons are formed. Here, the whole area of the brain is reorganizing itself to compensate for the loss of function, right? And this mainly happens in the cortical and sensory areas, allowing individuals to regain some lost abilities. So sensory areas are those areas which is responsible for your sensory functions, your sense organs related. All the information coming in from the sense organs are taken into the sensory areas. Cortical areas we'll see in the video where I'll be telling you about structure of brain. Okay, so here your brain, if some function is lost in this particular area, the whole of the rest of the area the nearby areas, they'll reorganize themselves. And how do they reorganize? Either by forming new connection between the neurons. Whatever neurons were severed or damaged or lost, they'll be forming here, they'll form new connections with the old existing neurons or strengthening the existing ones. Either they form new connections between neurons or they strengthen the existing ones. The third measure is adult neurogenesis. What is adult neurogenesis? Formation of new neurons. So if the damage is in hippocampus or cortical area, not cortical areas, olfactory bulb, new neurons will be formed to make up for the damage. And then fourth part is collateral sprouting. We saw what is collateral sprouting, right? So all these are methods by which a whole area of a brain reorganizes to make up for the damage. I hope you understand the difference between neural regeneration and neural reorganization. Neural regeneration is about a single neuron, while neural reorganization is about a part of the brain, the whole part reorganizing itself. All right. So yeah, again, we have covered neural degeneration, we have covered neural regeneration, we have covered neural reorganization, and the last we have neural recovery. Let's see what it is. Recovery involves a combination of brain's inherent ability to adapt and heal and external interventions like rehabilitation and therapy. Okay, so recovery can be physical, where neurons are regenerated, right? New reorganization happens, all of those, or it can be due to external interventions like rehabilitation and therapy, where your brain is provided a stimulating environment, right? What happens when you are getting a stimulating environment? Your brain is able to heal quickly. Okay. Physical exercises and cognitive exercises help in recovery of nervous system damage. Okay. Cognitive reserve. This is a concept that you need to know, not just for the exam, but also for your own healthy brain. Okay. It refers to the brain's ability to compensate for age-related or disease-related changes by using pre-existing cognitive processes or compensatory mechanism. Don't go with the bookish language. I'll tell you simply what this means. This means your brain is able to compensate for the age related or disease related changes if you already have a pre-existing information about it, or maybe your brain has always been active and neurons were always highly functional. And when are neurons highly functional? When there is more learning or more activity where the neurons have to be involved, right? So some individuals who are educated or intelligent, they are shown to be able to tolerate more brain damage and maintain better function. Why? This is due to the differences in how their brains process information and adapt. So this is like, more you use your brain, more active it is. More active it is, more neuroplasticity will be there, which means even if there is a damage, your brain knows that okay, I need to fix this because it's of use, it's highly used. Okay, I use my brain highly, so I need to fix the brain. That's how the theory works. And if you're not using your brain to study something, learn a new skill, or do better problem solving, if you're not using your brain at all, the neurons would not be firing as it should. And when a neuron is not being used as it should, it sits and degrades. And brain will also be like, okay, anyway, we are not using it. Why should I even bother to fix it? Right? So this should not be the case. Make sure you remain active, read a lot, or write a lot, or learn a new skill. Keep exercising your brain so that all these damages which might be due to age or due to any disease, the brain will be like trying to fix it by itself. All this will happen when the brain remains active and you keep it active. So keep learning. That's the best way. All right. So yeah, this was all about neuroplasticity. I have some previous year questions here which came from this topic. The latest one was June 2023 and it was a part question with two parts. Write short notes on the following 200 words. First was neural conduction, which we have already seen a previous video. Second was neuroplasticity. In June 2022, there was a 10-mark questions which said explain neuroplasticity of the brain, where you have to write all these parts. Whatever we have learned today, you have to write everything if a 10 marks question comes for neuroplasticity. December 2023, the same question but in a more specific manner. Explain the neuroplastic responses in neuroplasticity. What were the neuroplastic responses? We have learned 4 of them, like neural degeneration, regeneration, neural reorganization, and recovery. Alright, so that are the 4 neuroplastic responses for 10 marks. So with this, we come to the end of our first block and second unit. Make sure you let me know if you understood everything by liking the video, and if you have any doubt, anything you want me to take, just let me know by putting a comment, or you can drop an email to me. All right. So this is me, Dr. Swapna, signing off. Keep learning. Take care. Bye bye.